everyone, I am Rosandrella Salahay and I am going to tackle about the protein synthesis, which is all about the translation. So keep on watching.
What is a translation? Translation is the process of translating the sequence of a messenger RNA molecule to a sequence of amino acids during protein synthesis. The genetic code describes the relationship between the sequence of base pairs in a gene and the corresponding amino acid sequence that it encodes. There are components of the translational apparatus. One is the tRNA structure and the amino acid composition, which is the basic features. Next is the ribosome, which is the prokaryotic ribosomes and components. Then the eukaryotic ribosomes and components, the functions of ribosomal units. Then the mRNA and the genetic code, features of prokaryotic and eukaryotic mRNA. Genetic code and its features, wobbling and the genetic code. The amino acid activation and tRNA loading, specificity and fidelity of amino acylation reaction. Then the protein synthesis in prokaryotes, then the initiation complex and initiation factors. The initiation, elongation, and translocation, termination of translation, antibiotics, and prokaryotic protein synthesis. Then lastly is the eukaryotic translation, which is the eukaryotic initiation factors and initiation complex, initiation, elongation, termination, then the diphtheria toxin and its effects, and the iron and globin synthesis. Translation is also the process in which a sequence of nucleotide triplets in a messenger RNA gives rise to a specific sequence of amino acids during the synthesis of a polypeptide chain or protein. So the translation process occurs in cytosol and require the involvement of protein synthesizing units, the ribosomes, mRNA, tRNA, amino acids, amino acyl RNA synthetase, and several other proteins. During translation, an mRNA sequence is read using the genetic code, which is a set of rules that defines how an mRNA sequence is to be translated into the 20-letter code of amino acids, which are the building blocks of protein.
The code is a set of 3-letter combinations of nucleotides called codons, each of which corresponds with a specific amino acid or stop signal.
Translation occurs in a structure called the ribosome, which is a factory for synthesis of proteins. So the ribosomes has a small and a large subunit and is a complex molecule composed of several ribosomal RNA molecules and a number of proteins. Translation of mRNA molecule by ribosomes occurs in 3 stages: the initiation, elongation, and termination. During initiation, for the translation to begin, the start codon 5' AUG must be recognized. This is a codon specific to the amino acid methionine, which is nearly always the first amino acid in a polypeptide chain. At the 5' cap of mRNA, the small 40S subunit of the ribosome binds. Subsequently, the larger 60S subunit binds to complete the initiation complex. The next step, elongation, can now commence. While in the elongation, the ribosome has 2 tRNA binding sites, the P site, which holds the peptide chain, and the A site, which accepts the tRNA. While methionine tRNA occupies the P site, the amino acyl tRNA that is complementary to the next codon binds to the A site using energy yielded from the hydrolysis of GTP. Methionine moves from the P site to the A site to band new amino acid there, and so the growth of the peptide has begun. The tRNA molecule in the P site no longer has an attached amino acid and so leaves the ribosome. The ribosome then translocate along the mRNA molecule to the next codon again using energy yielded from the hydrolysis of GTP. Now, the growing peptide lies at the P site and the A site is open for the binding of the next amino acid tRNA, and the cycle continues. The polypeptide chain is built up in the direction from the N-terminal to the C-terminal or the final amino acid. Then the last stage of translation is the termination. One of the three stop codons enter the A site. No tRNA molecules bind to this codon, so the peptide and tRNA in the P site become hydrolyzed, releasing the polypeptide into the cytoplasm. The small and large subunits of the ribosome dissociate, ready for the next round of translation. Meaning, the termination occurs when the ribosome reaches a stop codon, the UAA, UAG, and UGA. Since there are no tRNA molecules that can recognize these codons, the ribosome recognizes that translation is complete. The new protein is then released and the translation complex comes apart. Importance of translation to the body. Translation is very important in the process of making proteins. Without translation, your body would have no possible way to make proteins or function. Proteins allow your body to do everything. Translation is very important also because it converts a sequence of nucleotides from DNA into a sequence of amino acids to build the desired protein. This process is essential for life. This can be found in all organisms, in eukaryotic and prokaryotic.
caught in the crossfire.